Today we're talking about Salmonella, one of the most important enteric pathogens in both veterinary and public health. This is a genus that crosses species boundaries, causes everything from mild diarrhea to severe septicemia, and plays a major role in food safety. By the end of this session, you'll have a clear understanding of how Salmonella behaves in different animal species, how it spreads, how to diagnose it, and what makes it such a persistent challenge in both clinical and agricultural settings. Let's get to it. Salmonella are gram-negative rods and facultative intracellular pathogens, meaning they can survive inside macrophages. That intracellular lifestyle is a big part of why they're so good at causing systemic disease and why they can persist in carrier animals. There are more than 2,500 serovars, but only a subset are major pathogens in animals. Many of these serovars are zoonotic, which is why Salmonella is such a big deal in food safety and public health. Most of what we deal with in veterinary medicine falls under Salmonella enterica. Within this species, we divide serovars into two broad categories. Typhoidal serovars are host adapted. They cause systemic disease in one primary species. A classic example is S. dublin in cattle. Non-typhoidal serovars have a broad host range and typically cause enteritis. These are the ones we see in outbreaks linked to food, water, or environmental contamination. Serovars tend to show up in different species. In cattle, S. dublin is the classic host-adapted serovar. It causes systemic disease, pneumonia, and chronic carriers. S. typhimurium is also common and tends to cause enteritis. In horses, S. typhimurium is the big one and is notorious for causing severe colitis in hospital outbreaks. In poultry, S. enteritidis is the major foodborne pathogen, while S. gallinarum and S. pylori cause host-adapted systemic diseases. Pigs get S. choleraesuis, which causes septicemia, and S. typhimurium, which causes enterocolitis. Reptiles can carry a wide range of serovars without showing clinical signs, a major zoonotic concern. Salmonella is typically transmitted via the fecal-oral route. Once ingested, it invades the intestinal epithelium, especially through M cells in the Peyer's patches. Inside the host, Salmonella can survive and replicate within macrophages. This ability to hide inside immune cells is what allows some serovars to cause systemic disease and establish chronic carriers. The inflammation triggered by Salmonella is what leads to diarrhea, often watery, sometimes bloody, depending on the severity of mucosal damage. Signs vary by species and serovar, but the big ones are diarrhea, fever, and dehydration. Young animals, calves, foals, piglets, are at higher risk for septicemia because their immune systems aren't fully developed. In cattle and horses, Salmonella can also cause abortion, either from systemic infection or from fever and endotoxemia. Some animals become chronic carriers without showing signs, which is a major challenge for herd management. S. dublin is the star of the show. It's host adapted, meaning it's really good at infecting cattle and causing systemic disease. Calves can develop pneumonia, septicemia, and high mortality. Adult cattle may show fever, diarrhea, or abortion. Chronic carriers shed intermittently, often without clinical signs, and can contaminate the environment or milk. This is a major food safety concern. Horses with Salmonella often present with severe acute colitis. They can have profuse watery diarrhea, high fever, and signs of endotoxemia, injected mucous membranes, tachycardia, and shock. One of the biggest concerns in equine hospitals is nosocomial outbreaks. Salmonella can spread rapidly through contaminated stalls, equipment, or even staff clothing. Strict biosecurity is essential. Poultry, Salmonella is both a production disease and a major food safety issue. Salmonella enteritidis can contaminate eggs internally, which is why egg safety programs are so important. Salmonella gallinarum and S. pylori cause systemic diseases that can lead to high mortality in young birds. These are host adapted and can be transmitted vertically from hen to chick. This typically starts with fecal culture, but PCR is increasingly used because it's faster and more sensitive. Serotyping helps identify which serovar you're dealing with, which is important for epidemiology and outbreak investigations. Environmental sampling, bedding, water troughs, feed is essential in herd outbreaks or hospital settings. The cornerstone of treatment: fluids, electrolytes, anti-inflammatories, and nutritional support. Antibiotics are controversial. They may be indicated in septicemic animals, but routine use in enteric cases can prolong shedding and contribute to resistance. Treating carriers is generally not recommended. It rarely eliminates infection and can worsen antimicrobial resistance. Prevention is all about reducing exposure. Good hygiene, clean water, proper manure management, and rodent control all help reduce environmental contamination. Feed contamination is a major source of Salmonella introduction, especially in poultry and swine. Identifying and managing carrier animals is essential in cattle and horses, where chronic shedding can maintain infection in a herd. Salmonella is one of the leading causes of foodborne illness in humans. Undercooked poultry, eggs, and raw milk are major sources. Reptiles are another important reservoir that can shed salmonella even when they look perfectly healthy. Young children, elderly individuals, and immunocompromised people are at higher risk for severe disease. Salmonella is a major enteric pathogen with significant impacts on animal health, herd management, and public health. Understanding species-specific patterns, recognizing the role of carriers, and implementing strong biosecurity measures are key to controlling the pathogen. And because of its zoonotic potential, Salmonella is one of the most important bacteria at the intersection of veterinary medicine and food safety. If you found this video helpful, please like and subscribe. Thanks for joining, and I'll see you next time.